Hi guys, this is Miss Polifka. So now we're entering into our stream analysis lab, and for this lab you're going to be writing your lab report. So you get 2 weeks to do this, and after the first week you can turn in your rough draft to me by the due date that's in your Blackboard, and I will give you feedback, all right? And you will be able to edit that and then submit it to me again. So in this first video, I want to talk to you about the test equipment and the stream analysis labs. I want to kind of give you an idea about what's going on in these labs and what you would be doing if you are taking in-person labs. The second video for the stream analysis lab will focus on how to write your lab report. Often scientists want to understand more about the state of something. They may be looking to gather data from rocks or soil samples, could be a stream or the atmosphere. In order to test these parts of our ecosystem, we often need to use specialized testing equipment. In this lab, we'll be looking at tests for nitrogen, phosphate, and dissolved oxygen. Now, before we talk about how to test for these things in our local water, we first need to understand why we test for these things. Why are they important when determining and discussing water quality? So let's go ahead and look at each of these factors in turn. In order to understand why nitrogen levels in a body of water is important, we have to look at the nitrogen cycle. You can see a simplified picture of this cycle on page 97 of your lab manual. Nitrogen is an essential nutrient for life and changes forms many times throughout its cycle. Now, there's a ton of nitrogen in our atmosphere. Two nitrogen bond together and form a diatomic nitrogen, or N2, and makes up the majority of our atmosphere, about 78%. But nitrogen in this form is not readily available to plants and animals to use. It has to be changed into another more usable form. We call this fixing. Now, this happens through a number of biological processes that we don't have to worry about too much for this class. Just know that nitrogen becomes fixed and it can form ammonium, NH4. It can form nitrates, NO3, or nitrites, NO2. In our waters, if nitrogen levels are too low, there's not enough food for plants and animals to grow and survive. If nitrogen levels are too high, algal blooms can occur. Algae are plants, and with lots of food to grow, blooms can occur. This is bad for our water systems because algae also uses the dissolved oxygen in the water and uses up so much of the oxygen that other plants and animals cannot survive. You will explore more about acceptable nitrate levels in your lab. Phosphate is another factor that affects the health and our aquatic ecosystems. Similar to nitrogen, phosphorus also has a cycle through our environment before it enters our waters. Much of our phosphorus that gets into the water comes from weathering of rocks and runoff from farms. Phosphates that enter the water are also used as food for aquatic plants and animals and can also lead to algal blooms when high concentrations are present. The last water test in this experiment is dissolved oxygen, or DO. DO is essential for life to thrive in aquatic systems. Fish still breathe air, but they don't do it through our atmosphere. They do it through filtering the dissolved oxygen out of the water through its gills. The amount of dissolved oxygen in water is dependent on two main factors: temperature and water flow. Colder waters have the ability to hold more DO than warmer waters. Moving waters also tend to have higher dissolved oxygen levels, as moving water pushes small amounts of oxygen from our atmosphere into our water. Looking at this waterfall, you can imagine just how this might occur.
Okay guys, so now we are at test point B. All right, so if you look that way, right over there is test point A, and behind me over here, this is test point C. All right, so that's where you're getting your 3 sets of data from. So here at test point B, what I'm going to start to do is I'm going to go ahead and begin by collecting a water sample, but it's not going to be just water, right? We want to get some of those living organisms in here, the micro and macro organisms. So what I'm going to do is I'm going to come out here and I'm going to kind of scoop some of this in. So now we have some suspended sediments, and then I'm just going to add some more water. All right, so that's going to go ahead and be my sample that we're going to test for the micro and macronutrients, as well as the nitrates, the phosphates. So what I can also do with this is I can use my pH meter to take the pH. Okay, and I can also get the temperature of the stream using just a regular alcohol thermometer. Okay, so you're going to be getting the temperature because dissolved oxygen is temperature dependent, right? So refer back to our lecture earlier about dissolved oxygen if you don't remember. Okay, so after we take those measurements, we can take a few other measurements as well. Okay, so this here that I have in this hand is a flow meter, and I can get over into the flow of the stream and insert it in here, and I can test the flow. I also brought a meter stick with me, as well as a 50-foot tape measure. All right, so the meter stick we would be using to measure the depth of the stream, and then we would need a second person to come over and use the measuring tape to measure the width of the stream.
So take a little bit to write your methods, okay? You're really going to have to imagine what you would be doing when you go to the stream in order to write your methods and procedures. All right, so that's the end of this video. Go ahead and click on the second video so that you can learn about how you're going to go ahead and write your lab report.